So thank you again for attending this soft tissue sarcoma session dedicated to ultra-rare subtypes and hereditary disposition. It is my honor to co-chair this session with my American colleague Brandi Felser. Sarcomas are, by definition, all rare cancers whose optimal care require rapid access to expert MDT in order to ensure consistent and optimal care and treatment. Yet, despite these now pretty well-known recommendations and some improvements in their management, patients' pathways still remain very heterogeneous, depending on the regions and the centers patients will be diagnosed. Among these rare cancers, sometimes arise ultra-rare subtypes of sarcomas with very complex profiles, whose diagnosis and management become even more challenging both for patients and physicians. It is my honor and privilege to welcome Dr. Silvia Stacchiotti, medical oncologist from the Istituto Nazionale dei Tumori in Milan, to tackle this very specific topic. Silvia, it's up to you.
So thank you very much again for being here and listening to what we are going to talk. This is going to be somehow the second part of what we started discussing before. And we are focusing on ultra-rare sarcomas, not because they are more important of rare sarcomas or sarcomas in general, but just because they help highlighting even more all the problems that we are facing when treating patients with sarcomas and developing new treatment for patients with sarcoma. So the definition of ultra-rare sarcoma is one or less, it is of a sarcoma with an incidence of one or less than one case per 1 million inhabitants per year. So the definition is based on incidence and was agreed by a group of a representative of experts in the area of sarcoma in 2020. Why we felt the need of a threshold to refine ultra-rare sarcoma? The reason was that we knew that some of the, the reason was that compared to early 2000, as built up yesterday mentioned, there was a paradigm shift. So at the beginning of my career, all sarcoma were investigated in the same study for any new drug. And this happened for anthracycline, this happened for pazopanib, this also happened for trabectedin. But over the last 20 years, this has changed completely, because what we realize is that every sarcoma is a different entity. So everything related to the biology, the natural history, the prognosis, and the activity of systemic drugs is different if we compare one sarcoma to another. And this makes it very, very difficult to develop new treatments, because of course, if you gather all sarcomas, the numbers are low, but not that low. If instead we want to develop a drug for each sarcoma type, the number of patients that can be treated with an investigational drug become smaller and smaller. And we understood over the years that there are histologies where, for statistical reasons and numbers, randomization is not feasible. Yesterday, the guy from the NCI spoke about alveolar sarcoma and forgot to mention that there was a study open at NCI that tried to compare sunitinib versus cediranib that basically took more than 5 years to include 20 patients, and at the end, the company was not interested anymore in the drug and in the disease. So, on this basis, to try to have a better understanding of what are the histologies among sarcomas in which randomization is really difficult, if not impossible, we decided to call under the umbrella of the Connective Tissue Oncology Society a representative of all sarcoma experts and say, according to you, what is the incidence cutoff below which randomization is really tough or almost impossible or impossible? And so looking at the WHO classification for sarcoma, for uterine tumors, because we have sarcoma split everywhere, and the last incidence data that were available, the community agreed upon this incidence cutoff. You must be aware that only for sarcoma we have a definition of what is to be considered ultra-rare. Now we talk about ultra-rare cancers, but this definition is not available. There is not an agreement on what type of sarcoma, what incidence threshold has to be used to call a cancer an ultra-rare cancers. The only other area where there is a definition for ultra-rare tumor is pediatric oncology, where they have selected, they have chosen the 2/1 million incidence cutoff. When we had this discussion, the majority said, no, 2 is too high. For example, with angiosarcoma, with chondrosarcoma, the disease is rare, but we can still randomize. So 2 is not the good number for us. We have to go below. And so we ended into 1 or less than 1 per 1 million inhabitants per year. Then looking at the WHO classification, the 2021, we found, and we also were surprised, that the majority of sarcoma histologic types are ultra-rare. They account for 20% of all bone sarcoma and all soft tissue sarcoma. By chance, the proportion is the same in the two groups. And they are approximately 75 different tumor types. For example, solitary fibrous tumor, angiosarcoma, chondrosarcoma are not ultra-rare. And the proof is that we have randomized study, meaning that these kind of studies are difficult but can be done. While if you go through all the whole list of ultra-rare sarcomas, the only histology where randomized study were ever conducted is alveolar sarcoma, the one I was mentioning before, with 2 studies that, as I said before, took so long that at the end had positive results, but the company was not interested anymore in considering the approval of the drug. Then, what is also important to know is that the new WHO classification that is currently under discussion, the pathologists are meeting right now this month, will have approximately 10 new entities, meaning that we will face in a year, in a book that will list 10 new sarcoma types that will be all by definition ultra-rares because they get out from other groups like UPS, not well-defined sarcoma entity. And you can imagine that at the time this diagnosis will be made by our pathologists as physicians, when we discuss, we will have the patient in the room, we will be like, I don't know what to say about your tumor. Because the literature around these entities is 3 cases, 5 cases, 10 cases, with almost no data on systemic treatment. And why I want to highlight that? Because we must be aware that even if we feel the urgency of providing the good consultation, sometimes we simply cannot give any kind of, better than general feedback on the specific disease. This makes retrospective data so important because to build prospective evidence on ultra-rare and especially new entities takes time. I need to set up a registry, I need to set up a study, I need to collect the cases, and the cases will take at least 5 years, to show what is the prognosis. So if we want to have something that can be used now, we must go back to our databases, review cases in which the diagnosis is similar or suggestive for the entity I'm searching for, and see what that databases, those data that are in our databases can tell me about the history of that tumor. And this will be, very important to design studies, even if they are retrospective data, and also to design prospective data collection. Because, if I want to start an observational prospective study or a prospective registry, I need to have at least some question in my mind. To do so, however, to make the best use of the data we have, of our retrospective data, and claim that they can be used also to design new studies, I need to be very, very good in selecting good data. And this is the reason why, again, with the same working group, we drafted this manuscript that tries to offer a list of the criteria that need to be followed when we want to collect retrospective data. Not every data we have in the database are good. Not every retrospective data we can collect are good. If we want to be reliable, if we want to be trusted even by the regulatory agency, we need to be able to show that the data that we are providing, even though retrospective, are good data. And here there is a list that is what we refer to when we now collect data into the push platform that we are going to talk soon. So again, to give you a sense of where we are, what agents are currently available for ultra-rare sarcomas? So how many of the 75 ultra-rare sarcoma we were mentioning before do have a specific drug approved for their indication? We have alveolar sarcoma and we have atezolizumab, which is approved only in the US. We have epithelial sarcoma and we have tazemetostat, again only in the US. Then we have, for the subgroup of GIS with PDGF receptor alpha, the A42V mutation, avapritinib, that is instead approved, I would say, globally. Then for IMT, inflammatory myofibroblastic tumor, we have crizotinib, that for some reason that I cannot explain, in Europe is approved only for pediatric patients. This is completely crazy, but this is the case. Then we have for entrapment-rearranged sarcoma, larotrectinib and trectinib, and only in the US in second-line repotrectinib. Then for RET-rearranged sarcoma, we have selpercatinib, again only in the US. Then we have nab-sirolimus for PEComa, again only in the US. And finally, we have eribulin for pleomorphic liposarcoma, meaning that out of 75 entities, we have less than 10 agents, and the majority approved only in the US, not available in Europe. So this is a shame, and we need to react and to find better solutions. This is very clear. So now, what is new for treatment of ultra-rare sarcoma that you can find in 2024-25? This is not meant to teach you about all ultra-rare sarcoma types, but they are somehow, in a way, some examples to show you where we are, and not to discourage you, but we cannot be advocate without being aware of the scenario. So again, let's go back to alveolar sarcoma. We mentioned that atezolizumab was approved based on a single-arm phase 2 study in which the drug was investigated in 52 patients only. 52 still is a huge number for alveolar sarcoma. And the drug, as presented yesterday, showed a very good overall response rate. So when we have a rare indication with a drug that can provide a high overall response rate by RECIST, a single-arm trial can be accepted. And this is not only true for sarcoma, it's true also for more common cancer. So this overall response rate of approximately 40% with a prolonged disease control. So what is important for us is not only that we see a response, but the response lasts for long, was considered good enough by the FDA to approve the drug that is now available in America. But the drug was never filed for approval in Europe, as I was mentioning before. And why so? Not because the company was scared about the negative assessment of the study, but because of the pricing. Because they knew that in case of approval, they would have had to rediscuss the price of the drug in all the other more common indications. So at the end, this would have been a loss for them, not a gain. And for that reason, and this is something we are seeing more and more, there is a discrepancy. So the drug is available here, but it's not available in Europe. If we don't change the rules, there is no future for patients. And also, what? Of course, I cannot give up. I work in Italy. I am the president of the Sarcomas Scientific Society in Italy. And so I feel that it's completely shaming that we don't have such a drug for this patient population. And with my group, we asked for the approval of the drug in Italy, and we achieved that. So what I want to show is that not only there is a disparity around the globe, but also within the same area, EU, you may have places where, in spite of no assessment by the EMA, the drug is available for patients, while in other areas it is not. This kind of, let's say, confusion is making the development of new treatments very, very much complicated. And there are cases in which patients contribute to the study. So usually Europe is a good place to run a study because there are a lot of patients, they are very well referred in a short period of time, but then at the end they have contributed to the study, but they cannot access the drug when the study is closed. And this is really terrible. And this is the case of a patient of mine that this is an early sarcoma case, is a nurse. She was diagnosed when she was 21 she started with brain metastasis. Then we discovered the primary tumor to the retroperitoneum with lung lesions. So at the time we had no drug approved, but we knew that sunitinib could work. So she was started in 2009 on sunitinib off-label, and she went on for 4 years. Then she progressed. At that time we were lucky enough that pazopanib was approved, so she was shifted to pazopanib, and she responded for 5 years, and then again she progressed. And at this point we had no other drug, so we tried an off-label drug, regorafenib, that she paid out of her pocket, and she responded again for a year. Then she progressed, and we had to go through doxorubicin, the only drug that is formally approved for the disease, that of course is not active, and indeed she progressed. And then we could by chance to get pembrolizumab. She again responded. Pembrolizumab is like atezolizumab, and she's now still on drug and free of progression after 4 years. So basically, she was started on active treatment in 2009. We are now in 2025, and she's still on treatment. She's working, she's doing well, almost all the time, thanks to drugs that are off-label. And many other exactly same kind of patient in my country cannot access the same kind of drug. Okay, another example is PEComa. PEComa is a tumor that often originates from the uterus. It is characterized by the activation of a chain of proteins related to the mTOR mechanisms of action. There are also patients with syndromic germline mutation. Based on this evidence, in 2010, Dr. Wagner tried sirolimus, which is an mTOR inhibitor, in a single case with a marvelous response that was published in JCO. And for 10 years, we could not convince any company making sirolimus or any other mTOR inhibitor to develop any of these drugs in PEComa, until now sirolimus was made. And only in the US, a study of nab-sirolimus in PEComa was conducted. This study, as expected, was strongly positive. It was a single-arm study only on 30 patients, so a small study. but the overall response rate was similar to atezolizumab in alveolar sarcoma. You see 38.7, almost 40%. And last follow-up of the study published in 2024 confirmed all the data and also the prolonged disease control. Based on these results, the FDA again approved the drug that is now available for patients, but again, the drug was never filed for approval in Europe. In this case, sirolimus can work, and retrospective data collected thanks to a global effort really show that the activity of sirolimus is comparable to the one of nab-sirolimus. And we are lucky enough that sirolimus is a very cheap drug now. So there is a chance for patients around the globe to be treated with an active drug, thanks to the fact that the drug is cheap, but not cheap enough everywhere. So this would be the perfect example of a repurposing project that we should be able to, push forward if the new legislation were approved. And this is why I think that in every part of the globe, we really need to, strongly support this big change. This is another point that I want to touch. In 2024, the community of experts also work around a manuscript on how to treat uterine sarcoma, including PEComa. This is another very important part of our job. Try to harmonize treatment of ultra-rare sarcomas and sarcomas in general is so much important because they are different diseases, and if we just leave to everyone to do what they want, we cannot offer the best service to our patients. So what is very important from the patient advocates is to ask for consensus guidelines on how to treat the disease, to try to harmonize the treatment, and also to highlight what are the open questions that need to be addressed by means of clinical studies. So this is one of the examples. We have other guidelines for EHE, chordoma, but many other sarcoma types are not covered by any kind of disease-specific guidelines. Then, I don't know how much time I have.
12 minutes.
12, okay. So another very nice example is epithelioid-hemangiosarcoma. Epithelioid-hemangiosarcoma is a translocated ultra-rare sarcoma characterized by two different fusions. It is interesting to know that probably in the next WHO classification, the less frequent fusion, YAPTAS, will be considered as a distinct entity. And this goes back to so if this happens, we will have to go back to all the data we have of EHE and separate the two tumors type and try to redescribe again what is the history of the tumor. Anyway, thanks to the global collaboration of the EHE Foundation, this tumor that when I started working in the sarcoma area in 2001, I remember very well, the first patient with EHE who entered in my clinic. I had nothing to say. And she was young, she was a super nice person. She died in 12 months, and I promised myself we need to improve what we know about the disease. And I'm a little bit proud to say that 20 years after, in spite of the disease being so rare, we know much more. We still need new drugs, but mostly thanks to the collaboration with patients, we were able to improve, because this is a disease of no interest for any of the company, at least until now. So we had no money arriving from any sponsor study, any company, and it was only thanks to the support of patient advocates that we could run a lot of studies and investigations. So for example, this is a study in a mice model that is important because we cannot run 10 tons of studies in this indication. We need to have pre-clinical data that can direct our our trials, showing that sirolimus in this case works much better than epirubicin. Then thanks to the collaboration with the patient advocates, we are probably identifying a cytokine that is related to the clinical presentation of the disease and could be used like a prognostic and predictor factor. And then there was the publication of a study conducted by the SARC showing that MEK inhibitor might have some activity, and the foundation is working hard to try to be able to access the data, because in such a rare tumor, we must be able to access the data, to dive into the data, to get the best we can in terms of information from the data of particularly academic study. Then we have worked a lot with sirolimus, and as I showed you before, we are discussing with the regulatory agency how to develop the drug in the disease. And thanks to the support also of the Sarcoma Foundation of America, we are now thinking to launch a global study that in a prospective fashion, observational study, will try to confirm prospectively the activity of the drug. This is very important because if this works, will be applicable to many other similar situations where otherwise for us it's impossible to improve our knowledge, to collect better data. And also, I'm very happy that what we have done over the last 15 years can be the good background for the new drugs that are coming out. So we have now new drugs, new compounds, in particular the TEAD inhibitors that maybe are more active when combined with other treatment. But what is very important is that thanks to the work that was done and should be done for every sarcoma, the agency do know the disease, do know the needs of the disease, already define with us with what kind of data they need. And at the time I go to a company and I'm asked, okay, what do you think about developing TEAD inhibitors in EHE? Is just a crazy idea? I can reply, no, it's a very good idea. Because there is a lot that you can make use of not to fail your project. Another tumor that instead is not in the same condition is intimal sarcoma. Intimal sarcoma is much more rare than epithelioid hemangiendothelioma. It is characterized molecularly by the presence of MDM2 amplification in, let's say, 50%, and EGFR amplification in the other 50%. The activity of conventional chemotherapy is really limited. The disease often arises from the heart, so the prognosis, you can imagine, is very bad. And thanks to the MDM2 amplification, MDM2 inhibitors, the same that were tried in liposarcoma, were tried also here, and they show to be active. And there is not only, but more than one publication. But the problem is that this drug failed liposarcoma, and there is no chance, no mechanism by which we can bring the drug to this really exceptionally rare tumor type. So again, we need mechanisms by which patients with this kind of very rare condition are protected in a way that if there is a drug, we should be in the position of developing the drug, you know? Otherwise, it's really very sad. And similarly, there are interesting news about immunotherapy in the same context. The Canadians published the activity of pembrolizumab in a few cases, but the company is not interested again. And I can understand it, because I see in Milano, we have very many, we see every day new patients, but I see probably 3-4 cases per year. How can I develop a drug when the numbers are so low without a global collaboration and a mechanism that can really support this kind of condition? So what I mean, there are good news in the literature, but this good news often just stop after the publication. Another tumor is desmoplastic round cell tumor. This is a disease that is characterized again by a translocation, affects a young patient population. Also, children and adolescents and young adults are affected. The prognosis, as you can see, is terrible. We have been using the same treatment we use for Ewing for years without knowing if this is a good idea or not. But there are data on pazopanib, and also data on immunotherapy, and also data on HER2 inhibitor. And this is something that was published this year. Again, how can we develop treatment, tried so many different kind of treatment in so rare tumor types? This is a good example in the disease. We were more lucky because we could test in a mice model the activity of trabectedin, lurbinectedin, and irinotecan. And we found also in a few patients that were treated more than off-label, because not only off-label, but this regimen was invented by the pediatrician, so not even very much regular. Anyway, the patient had a very good response. And similarly with trabectedin, we have seen disease control in patients that usually die in a few months for more than 5 years, like this case. So we went back to the company, PharmaMar in that case, who agreed to give us only the compound. Of course, running a study with the compound alone would have been very difficult, but thanks to the collaboration with patients who were in the drafting of the protocol and all the quality of life assessment and everything as of the beginning, we could get a very good grant by the charity, Rising Tide in this case, and the study is opening soon. Again, we will have a problem, because in case the study is positive, as we hope, I don't know what kind of further development we can think at the end of the study if the repurposing legislation does not go through. So running studies is difficult, sometimes it's feasible, but then what's next? Okay, probably you are tired. I don't think I have to go through all the examples I've prepared. But now I want to show you what instead we are doing within the PUSH platform. There is another ultra-rare tumors, low-grade fibromyxoid sarcoma, sclerosing epithelial fibrosarcoma. This is a spectrum of disease. A few years ago, meaning 3-4 years ago, I had to treat a neurologist, a very good guy, and I realized that in the literature there were only 5 cases published treated with doxorubicin. That's it. So I promised my colleague, who unfortunately passed away, that we would have done better than this. And thanks to the presence of this global collaboration of a center of expertise treating patients with sarcoma who are willing to put together all the data we have in the databases and following that rules I showed you in the paper about retrospective data, we could publish the first paper describing the history of the disease. This is very important to be aware of what happens to patients with this kind of tumor, to have something to base future study design, and also to be, in the WHO classification with some more element than the one we had in addition to the morphology or immunohistochemistry. And this was possible only thanks to this kind of collaboration. Also, the same kind of collaboration let us look at the data around the activity of chemotherapy, and particularly we found that immunotherapy, there are signals, and ifosfamide, there are signals, and on this basis, after retrospective data, we have now built a prospective observational study that will be conducted within the PUSH platform. The study is starting now, and what is also important of this model is that that when we have to launch a new initiative, we have the scientific part and the bureaucratic contracting part. And this second is terribly boring and time-consuming and costs a lot of money. So what is great is that we are trying to establish an agreement among all the participant institutions that are for this particular study were 35, and we foresee around 50 centers around the world, once forever, meaning that within that framework, using that platform, we will be able to design new protocols for each sarcoma type, ultra-rare sarcoma type, and of course we will need the IRB approval of the protocol, but without the need of signing a new contract every time. This is done once forever. And I really hope that this will speed up the data collection on all those histologies that are almost unknown to us. Similarly, we did with the pleomorphic, I'm finished. With after the low grade, we have done the same kind of exercise for pleomorphic and spindle cell rhabdomyosarcoma, and everything else will follow, the two abstract, and that's it. So this is just to say that histologies are so many that we cannot go too slow because patients are there. So we cannot say, no, we have not enough time, and the only way to do so is working together. This is a beautiful piece that you can find in Venice, and the sculpture is an American guy, is Anthony Quinn. I invite you to visit Venice and see the place, but even more to work together because we really need you, and thank you.
Thank you very much. I think we have time for one or two questions. Is there any questions in the audience?
I have a question. I wrote it down. One of the things that we spoke about is obviously the global data collection, which is why we're all here being connected globally, because as Dr. Stacchiotti pointed out, we really cannot make advancements unless, especially in these ultra rares, unless we're working together globally across the board. But one of the questions I have for you is you brought up a couple of topics of drug repurposing, and I know it's something that you're very focused on. And for the group, not that other research isn't important, but what do you see the role of drug repurposing in as a future for sarcoma treatments and therapies, and how big of a role do you think that will play? I personally think it might be, a really good avenue for us, but I would love to hear the expert opinion.
Yeah, I think that, one of the problems with sarcoma is that we are late. So we, that a drug starts being studied in phase 1 study. And usually these are basket study, not often open to sarcoma patients. But even in that case, sarcoma are so rare and all histologies are so rare that it's difficult that a signal is seen at that point. So often we understand that the drug works very late when there is no more interest from the company to develop the drug. But for us, it can be a very good option. And the only way is repurposing. And repurposing means two things. One is to try to work about the legislation, and the other one to work with companies to develop new formulation. In a way, nab-sirolimus is a good example of repurposing to me, because nab-sirolimus is exactly like sirolimus. It's only the route administration that is slightly different, and the company could build on the data of the activity of the drug in that indication that were already out there, and approve the drug thanks in a way to and this is an example that we are seeing even outside there. So also being able to talk with a company that can make, new formulation of the old drug that is of interest for us is an option. So I agree, repurposing is so important.
Any other questions?
You said that most of the available drugs for ultra-rare sarcomas only concern 10% of them, and that they were mainly approved in the US, not in Europe.
More, not many, but...
Okay. So what about the others? How do you manage them in Europe? these specific patients? How do you?
We don't manage. I mean, we have two different options. One option is to try to get the approval at local level, and every country has different rules. So, for example, I know that in France there is a facilitated access to off-label drugs for ultra-rare indications, while this is not the case in Italy. But, for example, in Germany, probably it's like in the US, all these drugs can be proposed to patients. So there are different, way to give the drug. But then there are areas where none of these drugs is, for example, East Europe is completely closed to any of these treatments.
Okay. And do you think that the current health policy in the US will have an impact on drug development or drug approval we might face in Europe?
I hope so. But this is for Brandi.
I think if I understand your question, where you said, do we think, can you repeat the question?
If the current health policy will have an impact on drug development, but also on drug approval, because we know that most of the drugs which are approved in Europe are firstly approved in the US. So do we have something to fear?
So I will be, yes. For lack of a better, I mean, look, if we're going to have an honest conversation about what's happening in the United States right now with some policy issues and things like that, I mean, it's the big sort of elephant in the room, and we are, especially in DC, so we can't ignore it. And I know from an SFA perspective, we work a lot in policy, and we're sitting at a lot of tables right now having these discussions of the future. But we don't know what's going to happen, right? I mean, from day to day, we don't even know what's going to happen with us. But here's the issues that we're having, and as we work with policymakers in the United States, and we sit, and for those of you who were here yesterday with Dr. Tapp talking about the Haystack Project panel, I was on that with him. It was him and I representing all rare cancers. There was about only 23 of us in the room, and it was hosted by Janet Woodcock, who is the former acting lead of the FDA. And we were talking about policy and writing, literally in the room, writing policy to change some of the regulatory pathways for approvals for rare diseases. But that being said, one of the things that's coming up is really specific examples of how people are being hurt by this, right? By all the changes and all the things that may be coming out. Everything could reverse. I mean, we don't really know what's going to happen, but here for patients, from a patient perspective, why this is so important. When you see PhD programs at universities having to be cut because of the uncertainty that's happening, the pathway, we already have limited researchers in sarcoma. They can't make a career out of it. There's not enough money already. So when we're cutting those programs, we're limiting our future pathway for research. That is going to hurt us. When industry is reticent to invest and move forward in drugs, when we already have these challenges right now we're adding more challenges because there's way more uncertainty. What's going to happen with that? We have labs closing down for research because the research dollars aren't there, and they're literally getting pulled out from underneath them. And then we also have, when you think about government funding, I know SFA is the largest private funder of research in the sarcoma community. We don't scratch the surface what government can fund. So when they're starting to pull back research funding, that is going to have an impact on further research. So all of these things, we don't know what's going to happen. And then, so yeah, there could be a huge impact. What happens in the US does impact others, but because so much happens here. But right now it is very uncertain, but I think we would be remiss not to have that conversation and really understand as patients, these are the real impacts that could happen should we continue to go down this road. We don't know for sure. It's all very new. I think everybody's trying to figure out. But when we're out there talking to people and people ask these questions and say, how is it impacting you? These are really clear examples of how it is going to impact us. And while I love the bigger cancers and we all need things, it does not impact them as much as it does us because we already have less. So when you reduce what we have, we basically are really down to nothing. So long answer to your question, but I feel like it's something that's been talked about in pockets, and I think it's an important thing for us all to address, even from a patient community. That's sort of the SFA's stance on it, and the information that we're getting from all of the areas and the policy discussions that we're having on multiple levels.
Thank you.
Sorry for the long answer, but.
Also, in Europe, the new pharma regulation, if approved, is gonna have a very big impact because, for example, there are incentives for pharmas, and one of the most important is that it foresees for rare tumors and pediatric tumors that after the EMA approval, they have to go only one step to get the pricing everywhere around Europe. And this is going to be the big difference. And also real-world data and data collected by patient, all of these is going to be relevant. The point is that we have been discussing, I mean, not me, but these pharma regulation for now at least 2 years.
And I was just in a meeting in Frankfurt talking about HTA and pricing in Europe, and that issue came up, so I have another question for you, and then I know we have to move on. But I think from the European standpoint, I know each country does it a little bit differently, but as you said, the legislation I have heard that there may be less incentive for pharma companies to go into Europe at all because of the pricing issue, and it can be different from each country, and if you negotiate, can you talk about that?
It's already like that. The reason why nab-sirolimus is not there, I believe, is this one. Because for atezolizumab, the problem was repricing everything, but nab-sirolimus is not there at all. But the cost for a drug that is developing a rare indication to go through each country, each agency at every country level is going to be huge.
Yeah.
And this is, of course, discouraging the investment in Europe.
Yeah.
Thank you.